Bead-based immunoassays, read by flow cytometers, have become an industry standard. Despite the growing capabilities and sensitivities of bead-based assays, beads and cells are typically analyzed separately. Multiple analytical instruments are often used to obtain data related to cell health, function, and type. Combining data together from separate instruments and samples is tedious and inefficient. With IQ Advanced Flow Cytometry, several individual assays can be collapsed into a single multiplexed assay. The IQ Phenotype and Function Kits allow for cells and bead-based assays to be combined into a single well to simultaneously assess cytokine secretion, cell health, and immunophenotypes. Get more insight from less sample with multiplexed analysis. More data, more insight in a single well. Integrated and concurrent data analysis leads to a streamlined workflow, alleviating the need to export data from multiple analytical instruments. This improved efficiency, along with better data coherence achieved with single-source sample analysis, makes identifying trends and hits that meet specific selection criteria quicker and more robust. Pairing efficient data analysis with comprehensive profiling and novel visualization tools, IQ Foresight software meets the demands of high-throughput drug discovery. Here we highlight the powerful analysis of IQ software for profiling a kinase inhibitor library using the IQ Human T-Cell Activation Kit. Profile maps were generated using the Panorama feature in the IQ Foresight software. The Panorama feature enables the instant analysis and reanalysis, if necessary, of multiple plates, even if run on separate days. Each of the desired metrics have user-definable thresholds, allowing for precise identification of wells of interest. These visualization tools provide simple insights into how the various kinase inhibitors impact different T-cell activation metrics. The need to rapidly monitor immune cell function is addressed by an optimized, high-throughput multiplex flow cytometry assay using the IQ platform and the IQ Phenotype and Function Kits. We have available kits to characterize T-cell biology, as well as immune cell killing kits. This figure illustrates one of many IQ Phenotype and Function Kits, IQ Reagent Kits, and the IQ Advanced Flow Cytometry platform, turning complex science into meaningful insights.